this video we will discuss about this induced proximity at the cell surface, how this strategy can be used for different objectives by the researchers. So first we will understand what does this induced proximity means. Induced proximity means at the cell surface level the proteins either come in contact with each other to trigger some desired pathways such as degradations, inhibit signaling, or activate signalings, or helps in the cell-cell interactions. And those examples are shown in this first figure, where we can see that when this reagent interacts with the two cell surface receptors, it might cause either degradations, or the receptors are or the reagents are designed in a way that it might inhibit the signal downstream signalings, or it will activate the downstream signaling. And in some terms this reagent helps in the protein transfers or cell killing. We can see that thing in the cell cytotoxicity assay, where the these reagents act as a motives, so that the immune cells get trained against those motives and then can generate the antibodies against those motives which are predominantly found on the cancer cells or disease cells. So these are some of the examples by which this induced proximity at cell surface can be used for the desired functions. And some of the details functions, some of the slightly more detailed analysis or slightly more detailed informations are shown in the bottom figure. So in the first figure it shows the example of these lysosomal degradations. So how this degradations occurs, that is shown in this panel A on the top figure, is that the receptors will take the proteins into the lysosomes for degradations, and this can done for the target proteins. So one of the way by to degrade these target proteins is through these lysosomal degradations, and other way is through this E3 ubiquitination. So in this case the target proteins, once it interact with the receptors, it will trigger the downstream signalings in a way that will cause ubiquitinations formations, or E3 ligase will form on these target proteins, and this target proteins then with the polymerized E3 ligase will form as signals towards ubiquitinations, and that will cause the proteasomal degradations once those target proteins reach to these proteasomals, and these things usually occurs in the cytosols. And some of the examples of this lysosomal degradations are shown in this panel B, where we can see that this LYTEC9 is one of the reagents that can be used to target these targeted proteins at a cell surface. And other one is this ASGPR, LYTEC15, and KINTEC16. These are some of the commonly used antibodies or reagent to target degradations of the specific proteins. And through the other strategies, that is ubiquitinations, there are also designs some antibodies such as APTEC10, PROTAB11, RULER 21, and these will trigger degradations based on the proteasomal ubiquitinations. So how they decided which one of the strategies to be used is depend on the tissue specific degradation. Is this one on depend on the tissue specific degradations, as which whether a particular tissues is more predominantly have this ubiquitination process or lysosomal degradations, or its natural degradations of the uh, proteins, such as proteins that are not able to form proper three-dimensional structures. So if the degradations need to be occurs by lysosomal degradations, then this strategy is used, that is the target proteins interact with the one of the antibodies that will cause this target proteins to reach to the lysosomals for its degradations. And in other case, if this targeted protein is not able to attack this targeted antibodies interact with this uh, off target tissues, or let me repeat this penalty. Yeah, so this is, we can consider it as targeted proteins, and in these targeted proteins the lysosomal degradations is not that common, whereas ubiquitination degradations is common. So if we use lysosomal degradations antibodies, then it will cause off target tissues and no degradations will occur, as the targeted proteins might not go to the lysosomals, as those pathways are not common in some tissues, whereas this ubiquitinations is if it is common in that tissues, then if we use this ubiquitination degradation method, then it will cause on target tissues to degrade. So this is one of the examples that shows that we need, before selecting this strategy, we need to understand the characteristics of this tissue or cells on which we are working, whether the ubiquitinations is the right strategy, or this lysosomal degradation is the right strategy. So I hope you understood this importance of this induced proximity for the researchers at the cell surface level, and how they use these strategies for getting their desired outcomes or to check their hypothesis. I hope you understood some of the basic understandings of this induced proximity at the cell surface. If you need to understand more about this topic in detail, you can go through the reference paper link that has been provided in the description. That's all for this video. Thank you for your time.